All right, we talked about the genetic code, and the genetic code tells us how to take an mRNA sequence and predict the polypeptide sequence that is produced. And now we want to talk about how the mRNA is produced. So we start out with DNA. So we know DNA is incredibly long. So each chromosome in humans, the largest is on the order of hundreds of megabases. Even the shortest is tens of megabases long. And so you have this huge long run of DNA. And from this DNA, you have to produce a relatively short mRNA. So in bacteria, when we talk about short, we're talking about something on the order of 1,000 base pairs. And so an mRNA is just a very small copy of the DNA sequence. So it's like taking a blueprint. So if you have a blueprint of how to build a house, you're just copying just one small part of that blueprint. For example, how to build a staircase, just a section of that. And so how is the cell actually able to know what to produce? And so, we know that a large number of mRNAs are produced for each gene. And in general, those mRNAs are almost identical. So they might not be exactly identical, but they are very close to identical in sequence from each other. So all of these start before the start codon, all of these end after the stop codon, and so you have sequences upstream of that and sequences downstream of that that are also included. So this sequence right here is often called the 5' UTR, and this sequence right here is called the 3' UTR. And the UTR stands for untranslated region. So, again, there's a start codon and AUG, and we talked before that there is contextual information upstream of that AUG that tells it that this is a start codon. So the mRNA has to include information not just at the start codon, but also some in front of it. And then it also has a little bit of information after that. So how does the cell go to the DNA and know where to start the mRNA and know where to stop the mRNA? And the idea is that the sequence is what determines this. So the sequence of the DNA includes regulatory sequences, promoter sequences, and terminator sequences. So the regulatory sequences, these are sites for the binding of regulatory proteins, and the role of regulatory proteins is to influence the rate of transcription. So these are found in a large variety of locations, and in one of the later chapters, two of the later chapters, we'll talk in detail about how these are used to control transcription. The promoter region, this is what is used to indicate where the mRNA should start. So this is a site for initial binding of an RNA polymerase. So the RNA polymerase is the enzyme that transcribes the DNA to produce the mRNA molecule. And it signals both this initial binding and then the initiation of transcription. So the polymerase comes here, it binds, and now it starts to produce this mRNA. And then as it moves forward, it continues to make more mRNA, and it does that until it reaches a terminator sequence. And so there's certain things in the DNA that indicate for the mRNA to stop, and then at this point it can release, and then you have an mRNA molecule. So the mRNA, when we look at the organization of the mRNA, so you have the ribosome binding sequence. So this is kind of similar. There's sequence there that indicates for the ribosome, which translates the mRNA into polypeptides. For that to initially bind, you have the start codon, which indicates this is where the start of the polypeptide is. This is the first methionine that gets incorporated. You have codons that are used to encode additional amino acids until it reaches the stop codon and specifies the end of the polypeptide synthesis. One thing to talk about is that bacterial mRNA may be polycistronic, and what that means that it can encode 2 or more polypeptides in an operon. So you might have something, a mRNA molecule that is long, but it actually contains 2 different ribosome binding sites. So one here, and then you would also have a start codon, a stop codon, a start codon, and a stop codon. And you can do this for 2 genes, you can do this for 3, 4, 5, we're going to get into this more. So bacterial mRNA is often polycistronic, which it means that this mRNA that produces is not just a single gene, but it actually contains multiple genes. And so the reason that's possible is that what transcription is essentially independent of translation. So the signals for the start of transcription and the end of transcription are completely different from the signals of the start of translation and the end of translation. And so, you know, come back to this slide after you've gone through these activities, after we've gone through the other chapters, and just remind yourself. It might make more sense what exactly I mean by that. But it's very important to remember that the signals for the start and stop of transcription are independent from the signals for the start and stop of translation. So gene expression requires base sequences. So the DNA strand that is used as the template is termed the template strand or the antisense strand. And the RNA transcript is complementary to the template strand. So the opposite strand is called the coding strand or the sense strand, as well as the non-template strand. And so the base sequence of this is identical to the RNA transcript, with the exception for the substitution of uracil and RNA for thymine and DNA. And then the transcription factors that initiate transcription, recognize the promoter and regulatory sequences to control transcription. And then the mRNA sequences such as the ribosome binding site and codons direct translation. So here, this is just kind of a visual indication of what we mean. So DNA is double-stranded, and only one of those strands can be used by the RNA polymerase in order to create an mRNA transcript. So in DNA replication world, we always had 2 DNA polymerase 3s in order to replicate each of the strands. But for transcription, you're only trying to do one of the strands. So you only have a single RNA polymerase molecule. So this is built of multiple subunits, but you have a single single super molecule that's the RNA polymerase. And so this has to actually be binding to one of these 2 strands. And it has to be reading the strand in a certain direction. So just like DNA sequencing, just like DNA replication, RNA is produced in the 5' to the 3' way. So we would have started transcription here, and then we are moving this way. So the strand that it is reading, the template strand, the antisense strand, is going from the 3' to the 5' direction. And so every single nucleotide that gets incorporated is complementary to that antisense strand. So that C causes a G, et cetera, et cetera. So the sense strand, the coding strand is the opposite one. So this goes in the 5' to 3' direction. So the sense strand is never bound by the RNA polymerase, but because of the complementary nature of this, it is almost exactly the same as the RNA transcript. So you'll see the T, the A, and the G, the G, C, C, and then the only exception is because RNA uses uracil instead of thymine, these T's get replaced by U. So the sense strand we can take and we can just change the T's to U's, and then we know exactly what the mRNA sequence is from that. But this is just terminology, so just make sure you remember that the antisense strand or the template strand is the strand that the RNA polymerase binds to, but it's the reverse complement of the mRNA sequence that is produced. The coding strand is not actually bound to or used by the RNA polymerase, but it has almost the exact same sequence as the mRNA that's produced. So we break up transcription into 3 different stages. So initiation, elongation, and termination. So this is pretty straightforward. And all of these steps involve protein-DNA interactions. So again, DNA sequence always matters. So whenever you think about how DNA works, you almost always just think about what is the exact sequence of the DNA. So in some cases, it's encoding protein, but in other cases, it's simply working in a way to allow proteins to bind it. So there's proteins that can recognize nucleotides. And so if you have a set of those nucleotides in the proper order, then the proteins can come in and bind to it. So an example of this is RNA polymerase. So RNA polymerase interacts and binds to the DNA sequences. So when we went over the chemistry of DNA of a nucleotide, one of the things I asked you to do was look at all the hydrogen bonds, all of the different parts of the atoms, all of the different atoms that that could create hydrogen bonds. So some of those hydrogen bonds were used to produce the double-stranded DNA, which allowed the A's to bind to T's and the C's to bind to G's. But you also should have seen that those nucleobases had other hydrogens and nitrogens that could also form hydrogen bonds. And so those other ones are what the proteins can go in and actually bind to kind of read what the DNA sequence is. So initiation, the process of initiation is when the RNA polymerase is able to bind to the promoter. So the first thing that needs to happen is the promoter must be bound to. You must melt the DNA strands apart, and then you must initiate and start the formation of an mRNA transcript. So for transcription, you don't need a primer. So this is one of the ways to distinguish between DNA replication and transcription, is that in order to produce an RNA molecule, you do not have to start with any sort of primer sequence. And so the start of the mRNA is just exactly complementary to where the promoter is. After initiation occurs, then the polymerase reads along the DNA and it produces mRNA in a 5' to 3' direction. So this end is the 5' end. What's going to be produced is the 3' end, and it just slides along the DNA in an open complex, incorporating complementary nucleotides to the template antisense strand. And then finally, this polymerase reaches a sequence that is the termination sequence, and we'll go over the mechanism of that, but that allows the RNA polymerase to disassociate from the DNA, end transcription, and release the completed RNA transcript. And so the last nucleotides, ribonucleotides that are incorporated are on the 3' end of the mRNA. So study of a large number of genes and study of where they are transcribed, what the mRNA produced, has led to the identification of certain key sequences that are upstream of the start of the transcript that are important for binding of the RNA polymerase. So before we get into this, we have to talk a little bit about terminology for how we name and number parts of the DNA that are nearby the initiating site for transcription. And so here I'm showing you this is the mRNA. So this right here is the mRNA that is produced. And this is the first sequence, the first nucleotide, ribonucleotide, occurs right here. And so we name this site the +1 site. So the +1 site is the first nucleotide, ribonucleotide, the first DNA nucleotide that encodes something that will end up in the mRNA sequence. So this is all DNA, this is DNA, and then the red is the RNA. And so that is called the +1 site. And then you would just move this way to do the +2 site, +3, +4, etc. So the first part of the DNA that is in the mRNA is called the +1. And now we just go backwards from that in the negative direction in order to name the nucleotides in the DNA that are upstream of the initiation site, the transcriptional start site. So the -10 sequence is approximately 10 base pairs upstream of the initiator site. The -35 sequence is approximately 35 base pairs upstream of that site. So none of this DNA right here is actually incorporated into the mRNA, but it is critical for the initial binding of the polymerase and the eventual initiation of the transcript and the start of the mRNA. So there is no base numbered 0, so bases preceding the start site are numbered in the negative direction. So most of the promoter region, so the promoter region is where the polymerase initially binds. Almost all of that is in the negative region. Sorry, this is some messed up animation, unfortunately. So these sequences, so the promoter may span a large region, but specific short sequence elements are particularly critical. So you can see we're only talking about 12 base pairs. So these 12 base pairs are pretty critical for regulating and allowing the initiation of the promoter site. So this part in the -10 region is called the Pribnow box, after it's discovered, and it is something like a TAT-AAT, and then the -35 region is TTGACA. So if we look at a bunch of genes that had been characterized, some of the original genes to be characterized, so these are the names of the genes. So there's the lac operon, lac-I, trip operon, etc. So these are all independent genes. They're all transcribed. This is a tRNA. And so if we look at the first base pair of the mRNA, this of course is right here, the initiator site. So most of the initiation has an A. And then if we look 10 base pairs upstream, so you can see how many base pairs are in between the start and what is the Pribnow box or the -10 sequence is. So in this case, there's 6 different nucleotides, 7, 7, 7, 7, 6, 7. So in general, there's 6 or 7, so there's some flexibility where one base can kind of go back and forth. But in general, they're almost always exactly 6 or 7 nucleotides upstream of that A. So this would be the -7, -8, -9, -10, -11, and -12. And so you see that the consensus sequence is TAT-AAT, but almost none of these genes have an exact match. So this one is TAT-GTT, this is CAT-GAT. So they're close. Typically there's only one base pair difference, maybe 2 base pairs that are different, but they're definitely not exact matches. And then the next one is the consensus sequence for the -35 region. So typically there's 16 or 17 base pairs in between this. And then you can see these are just real life examples of what that sequence looks like. So in general, there's often 1 or 2 base pairs that actually don't match the consensus sequence exactly. So this is a bit unfortunate from a bioinformatics perspective for our ability to predict the start of transcription. So it's actually still pretty hard for us to predict just using DNA sequence where the start of transcription will occur. We're much better at predicting the start of translation because of that rigid rule of having to use an AUG. So we can there's certainly programs that attempt to do this, but they are not perfect in any way. So often we have to experimentally determine the start of transcription because of that. But once you characterize it, if you look upstream, very often you'll find something that looks a lot like the consensus sequence. So the consensus sequence is just the most commonly occurring basis, and for many bacterial genes, there's also a correlation between the rate of RNA transcription and the degree of agreement. So the better they match this exactly, the more they are transcribed and the higher the mRNA levels are. So RNA polymerase is the enzyme that catalyzes the synthesis of RNA. In E. coli, the RNA polymerase is a holoenzyme that's composed of multiple subunits. So there's 2 alpha subunits, a beta prime, and an omega subunit. These form the core activities of the polymerase that actually produces the mRNA. And then there's an important accessory factor called the sigma factor. So all of these factors can bind together to produce kind of a super protein enzyme. And each of these subunits play distinct roles. So the one that's most important, the one that I really expect you to know for an exam, let's say, is the sigma factor. And the reason I want you to know that is because the sigma factor is what's key for recognizing that consensus DNA sequence. So the reason that the DNA has to have something that matches that sequence is that this omega factor, this omega subunit, is actually forming hydrogen bonds and allowing the binding between the RNA polymerase and the DNA, double-stranded DNA. So the sigma factor is what brings the polymerase to the promoter site, and then once that occurs and transcription can initiate, so we start to create this mRNA molecule, now the sigma factor's its job is done and it simply disassociates from the RNA polymerase. So that RNA polymerase still has all of these other factors within it that are producing the mRNA transcript. And so again, just to reiterate that, it's important to really think about protein is able to bind to DNA. So what we see here is part of the sigma factor. So this yellow is part of the protein, the sigma factor. And so this sigma factor has this helix turn helix. So this is a helix, which we'll talk again more about, another helix and a turn. But it's these amino acids within there that's actually recognizing what is the DNA sequence within the double-stranded DNA. And so again, this is because there's these atoms within the nucleobase, the nitrogenous base, that can be used for hydrogen bonding. And so in this way, the sigma factor is able to bind to the DNA and recognize the right sequence. So if the DNA sequence is not correct, then the sigma factor is unable to form those hydrogen bonds. But because it is, then it is able to, if it is the correct sequence or near enough the correct sequence, then it's able to bind and produce this complex and initiate transcription. So the RNA polymerase binds kind of loosely to the DNA. So how does initiation occur? So it kind of binds loosely, and then it starts to scan the DNA. And so initially the sigma factor is not able to produce those bonds and stabilize the RNA polymerase. But once it moves to the point where the sigma factor is complementary, not complementary, but in the sense of complementary nucleotides, but complementary into the sense of the amino acids can bind the nucleotides, then we have a tighter binding. And so now the complex can close together and initiate the initial transcription of the mRNA, and then the sigma factor can dissociate. The open complex formed by the action of RNA polymerase is about 17 bases long. And then behind the open complex, so that means that there's about 17 bases of denatured DNA, so where the double strands have been separated, and then behind the open complex, as the RNA polymerase moves along, the DNA just rewinds itself back into a double helix. And on average, the rate of RNA synthesis is about 43 nucleotides per second. So once elongation occurs, you're incorporating nucleotides pretty fast into each of these mRNA, faster than that. So there's not really much to know about elongation, but termination is when it ends. So termination is the end of RNA synthesis. It occurs when the short RNA-DNA hybrid of the open complex is forced to separate, and this releases the newly made RNA as well as the RNA polymerase. And so in E. coli, this is the bacteria that a lot of this fundamental work work was done, there's two independent mechanisms that are used for termination. So one is called rho-dependent termination and requires a special protein known as rho. And then there's rho-independent termination, which does not require this protein factor. So we're going to talk a little bit about both of them. So one of them is rho. So rho is this protein that almost looks like a circle. And so within this circle, the RNA is actually able to bind to the RNA and then kind of move along the newly synthesized RNA molecule. So it initially binds to a recognition site in the mRNA. And so once the rho protein binds to the site, it's able to move along the mRNA and try to catch up to the RNA polymerase that is synthesizing new, adding new nucleotides, incorporating new nucleotides into the mRNA. So eventually the RNA polymerase transcribes a region that forms a stem loop and then proceeds to the terminator. So what happens is that this this mRNA, if it has if the mRNA is able to form a stem loop, it pauses the RNA polymerase. So the RNA polymerase is no longer to go moving at 43 base pairs per second. And then at this point, the rho protein, because of this pausing, the rho protein is continue along and is able to move along. And then as the rho protein catches up, it eventually moves where the active part of the polymerase is, and it breaks apart the binding between the RNA and the DNA base pairs, and it releases the protein, releases the mRNA, and releases the polymerase. So part of what's important for termination to occur is the stem loop, and then part of what's important for this to occur is this binding site so that the rho can bind and then move along the mRNA molecule. So what is a stem loop? So this is kind of a visual depiction of what a stem loop looks like. So this is in the mRNA sequence. And so what allows this stem loop to occur is if there's complementary base pairs in the mRNA sequence that allows it to form double-stranded RNA. And so you'll notice that it's anti-parallel. So here is the 3' end. Here is the 5' end. So the directionality of the strand here is the 5' to 3' direction. But in this case, for this side, it's the opposite. So the 5' to 3', if we look at the directionality of it. And so you end up having anti-parallel bonding, which is necessary to form double-stranded RNA as well. And so this is possible if these nucleotides right here are all complementary to each other. So they can be any set of nucleotides, but one example of this is if these were all C's right here, so if you had a run of C's, and then before it you had a run of G's. So in this case, if you had a run of G's and a run of C's, then these are complementary to each other, and so they can actually form this double-stranded RNA. But they have to be anti-parallel, and then they have to be complementary to each other. And so this stem loop is important for terminating transcription in combination with the actions of the rho protein. So the rut site, that is the rho utilization site. So the rho protein is a helicase. It's a type of helicase that's able to move along the RNA molecule. There's also rho-independent termination. So this does not require the use of a protein, but again, it uses a stem loop. So again, we're seeing the formation of this hairpin. And again, this causes pausing of the RNA polymerase. So it's no longer able to zip along, zip through the DNA, read it, incorporate new mRNAs, but it actually starts to slow down. And so the sequence, so there's no rut site, but there's additional important sequence information that's necessary for this termination to occur. So again, we need the sequence that's part of this hairpin, and then we also need to have a run of U's or A's. And so the reason for that is just, if you remember, U's and A's have 2 hydrogen bonds, C's and G's have 3 hydrogen bonds. So these bonds, the U and A, is weaker than the bond between the C's and G's. And so if you have a large number of these U's or a large number of these A's, you end up having a much weaker binding in between the DNA and the RNA molecule. And so this weak base pairing is kind of similar to what happens when the Rho protein comes along and then just releases and splits the RNA and the DNA from each other. So it's just the action of the weak binding also allows for this to occur, and so the RNA molecule is released and the polymerase can then disassociate from the DNA, and then termination occurs. So that is rho-independent termination, and that's facilitated by 2 sequences in the RNA: the base, the hairpin, and then the run of AU base pairing. So finally, both strands of DNA can be used as template strands. So just remember that a gene has direction. So a gene can either go, so if this is the origin of replication right here, a gene can either go away, point away towards the origin of replication, or it can point towards it. And so that is completely determined by the promoter sequence. So the promoter sequence has directionality. So, if we look at the, if we go all the way back and just remind ourselves what the sequence is, so TAT, AAT. So so if you have it here, another possibility you can have is it in the opposite direction. So, this sequence right here, if you had TAT, AAT, then this would specify transcription in this direction. But if you had it on the opposite strand, then it would specify transcription in the opposite direction. So, you have to just remember where this consensus binding site is. Is it on the Watson strand, the top strand, or is it on the Crick strand, the bottom strand? And so if it depending on which one it is, it will drive transcription in opposite directions. And so if you look at the E. coli chromosome, the E. coli genome, you're going to find directionality for the genes. So there's going to be genes that are transcribed in both directions. All right, and so that is the end of transcription in prokaryotes. So just remember there's 3, so kind of the important features, there's 3 different parts of transcription. There's the initiation, there's the elongation, there's the termination. And just, to remember that the mechanism of that is specific to transcription. So what we described is how mRNA molecules are produced. What we're talking about has nothing to do with translation, and there's completely different rules for initiating translation of the mRNA into polypeptides.